Okay, so we know that we get more protein into the muscles that have been exercised. When we eat protein after our exercise, we get a bigger increase in protein synthesis, a slightly lower increase in degradation. And that if we have protein, it seems like it's going to improve our increase in muscle mass and strength after exercise. So what does protein do? And are there specific amino acids that we're really trying to get? And so this busy slide here, it's looking at the effect of leucine on mTOR activation. That's the yellow line here, and that's going to be on our left axis. And then we're also looking at protein synthesis. That's the blue line here. And what you can see in this experiment, what this group out of Penn State did, and this is Scott Kimball's group, what they did is they gave in this case rats 5%, 10%, 25%, 50%, or 100% of the daily recommended amount of leucine, just as a single little drink that the animals would quickly drink. And once they had drunk the leucine, then what happens is they're going to wait a small period of time, and then they're going to collect the tissue, and they're going to collect the tissue, and they're going to measure mTOR activity, and they're going to measure protein synthesis. Okay, and so what you can see here is that as we go from 5% all the way up to 100%, there's actually a linear progression in mTOR activity. So the more leucine you get, the more mTOR activity you have within the muscle. And so that's really important because that's showing us that leucine can specifically turn on mTOR within muscle. Okay, so what that's the most important thing that's telling us is that leucine is a direct trigger for mTOR activity. If we look at protein synthesis, however, it goes up, and then about 15 to 20, between 10 and 25%, it kind of plateaus. And so the maximal amount of protein synthesis happens between basically 10 to 25% activation of mTOR, or 10 to 25% of the daily value of leucine. So what could be the things that are limiting here? Well, they were looking at the effect of a single amino acid in leucine. So what's going to happen is you're building your wall, but now you only have one color brick for your wall. You're never going to be able to make you're never going to be able to make this pretty wall with all of these different colors in the very specific repeating pattern. You're not going to be able to do that because you only have the one color brick. So you're going to make a very plain wall, but you're also going to get to the point where you're all out of the other bricks, and that's going to happen here. Or it could be that what's happening is that even though you've got more and more mTOR activity, now something else is becoming limiting. Early on, mTOR is limiting protein synthesis, but it's possible that later on, something else is limiting protein synthesis. And a lot of people have taken this data to mean that there's not enough ribosomes anymore. So you can only make. So again, the analogy here is that if you have a factory and you have 3 machines to build your thneed, if you have one person, one employee, well, now you're getting only 1/3 of the amount. If you hire another employee, you can double the amount of output that you have. If you add another person, you can get another big increase in output. So you can get that big increase in output early on. But then if you've got 3 people working, it doesn't matter if you hired 3 more people. If you hire 3 more people, that's not going to do anything, because now instead of the number of people working the machines being the limiting step, now the limiting step is the number of machines you have. And so it could be that this is indicating that there's not enough ribosome mass to increase protein synthesis more, or it could be that there's not enough amino acids to build the protein. It's one of the two, and it hasn't really been finalized as to what is causing this plateau, but we know that mTOR activity goes up with leucine, protein synthesis goes up, and then it plateaus. All right. So now, how does this happen? And that's the paper that I gave you this time. for this presentation or this lecture is this paper from Sanic. It's out of the Sabatini lab. David Sabatini is one of the co-discoverers of mTOR, and he is the world expert in how mTOR is activated or is regulated by nutrients, so amino acids and sugars and other things. And so what they did in this really cool study is they took away amino acids for 50 minutes from the cells. And when they looked to see where mTOR was, mTOR is in green. It's everywhere throughout the cell, except in this area where that's the nucleus. You can see the DNA is right there. Okay, and then all they did is they fed amino acids for 10 minutes, and now every bit of mTOR is now in these little dots. And so that was really cool because it meant that mTOR changes its location when you get amino acids. So how does this happen now? Well, what they were looking at here is this Rab7, and Rab7 is a marker for the lysosome. And so what you can see is Rab7 is at the lysosome. Rab7 is at the lysosome when there's no amino acids. You add amino acids, Rab7 is still at the lysosome. And now mTOR is all at the lysosome. So that's interesting, sure. But then what does that tell us? Why is that important? Well, this part of the experiment is what tells us why that's important. If we look at Reb, again, Reb is the mTOR activator. You can see that Reb is outside of the nucleus in this area right here, and it's kind of spotted together with Rab7. In the absence of amino acids, it's in Rab7 positive cell location. When you add amino acids, Reb is still there. That means that it's kind of tethered to the lysosome. So now the interesting thing is that, remember, Reb is on the lysosome, TSC2 is there, and if we've done something and moved TSC2 away, now what you're doing with amino acids is basically you're bringing all of the mTOR to Reb. So what amino acids do is they bring mTOR to its activator, and what growth factors or resistance exercise do is they move the inhibitor of Reb away from Reb. And so now this is why these two things together, resistance exercise plus amino acids, give you a bigger effect than either one alone because they do two different things. They activate mTOR in two different ways. One moves the inhibitor of Reb away, and the other moves mTOR to Reb. Okay? So how does this work? How are we moving mTOR to Reb? Well, what the Sanic paper started, and he's had a number of other papers following up on this, is that what we've realized is that there's these small GTPases called the RAG proteins. And the RAG proteins come in a number of different flavors. And what we know is that it's a dimer. And they have to function as a dimer where RAG-C is bound to either RAG-A or B, and RAG-A is going to be bound to C or D. So one of A or B is going to be bound to one of C or D. We know that when RAG-AB is in the GTP-bound state, now we've got active mTOR. All right, so when we give amino acids, now what's happening is that a guanine nucleotide exchange factor is coming in. We're getting RAG-AB into the GTP-bound state. And when you fast, you're going to activate a GAP protein. So a GEF, remember, is a GTPase, it's a guanine nucleotide exchange factor that's going to kick out the GDP and bring in the GTP because GTP is tenfold higher in the cell. So once you kick out the GDP, it's 10 times easier for you to find a GTP than it is to find a GDP. So the result is you get GTP bound. In the fasting state, now we've got a GAP protein, a GTPase activating protein, which is going to break the GTP, help RAG break the GTP down to GDP and turn this back off. So in a fasting state, this GAP protein is always there and it always keeps the RAG protein in the off state. When we feed amino acids, now we're going to get rid of the GAP and now we're going to bring in the GEF. Okay? All right. So how is this regulated? Well, the first thing is let's work from the leucine side of things. So what does leucine bind to? And this is data out of David Sabatini's laboratory where there's this protein cestrin. And if you mutate two sites on cestrin, this is how much leucine can bind to cestrin. And what you can see is that cestrin 1, 2, or 3 binds to leucine quite effectively. If you mutate these sites right here to mutate the leucine binding pocket, now you see that you've eliminated all of the ability of leucine to bind. And so what does that do? Well, what does that do? That comes in and it works with this protein complex called GATOR2. And GATOR2 has 3 or 4 proteins, one of which is the WDR24. And when you have leucine, you go from 0 leucine to 20 micromolar leucine to 100 micromolar leucine. What happens is, if I pull down flagged WDR24, so if I pull down GATOR2, as I increase the amount of leucine there, I see less cestrin together with GATOR2. And so GATOR2 is binding to cestrin in a leucine-dependent way. So when there's leucine there, cestrin moves away. And WDR24 and GATOR2 can do what its job is. Okay, so how does this work? It works like this. So what you have is you have leucine, cestrin, GATOR2, and GATOR1. And the important thing to start with is that GATOR1 is the gap protein. That's the GTPase activating protein for RAG. And when it's here, when there's no amino acids, the GATOR2 cannot inhibit GATOR1, and GATOR1 is turning off the RAG-AB. And so it's always going to be in the RAG-AB GTP-bound state. When you bind leucine to cestrin, what cestrin normally does is cestrin inhibits GATOR2. And when you inhibit an inhibitor, that is an activation. So leucine comes in, binds to cestrin, and now cestrin doesn't bind to GATOR2 anymore. Okay? Let's say that again. Leucine, when it binds to cestrin, it removes cestrin from interacting with GATOR2, and now GATOR2 can become active. And when GATOR2 is active, it blocks the gap and prevents this moving back into the GDP-bound state. Okay? The GEF protein for this, for RAG-AB, is something called the regulator. Again, this is a nice name because it's the regulator of RAGs. So it's the regulator. And what the regulator does is it does a number of things. One, it keeps the RAGs at the lysosome. And 2, it can cause the RAGs to become GTP loaded. And it's always trying to GTP load the RAGs, but the GATOR1 protein is always trying to turn it off. So when leucine is present, cestrin moves away from GATOR2, and GATOR2 now blocks GATOR1 and prevents the turning off or the GTP ACE activity of the RAG-AB. Okay? I know it's complex. Go back through it a couple of times, you'll get it. RAG-AB, when it's GTP bound, what does it do? It binds to Raptor. And Raptor is what brings mTOR complex 1 into or together with the lysosome.